Welcome to the GlobalRPh YouTube channel, your trusted resource for clinical excellence and evidence-based medical education. The concept of the Hayflick limit, first elucidated by Leonard Hayflick in 1962, revolutionized our understanding of cellular aging. This seminal discovery revealed that human somatic cells possess a finite replicative capacity, undergoing a predetermined number of divisions before entering an irreversible state of cellular senescence. This fundamental biological constraint underpins the maximum potential human lifespan, estimated at approximately 120 years. The cellular replication limit was first observed in 1961 when Leonard Hayflick and Paul Moorhead demonstrated that normal human cells undergo a finite number of divisions. This groundbreaking discovery overturned previous beliefs about cellular immortality. It established the groundwork for modern gerontology and aging research. The Hayflick limit refers to the maximum number of times a normal human somatic cell population can divide. For human fetal fibroblasts, this ceiling ranges between 40 and 60 divisions in laboratory conditions. After reaching this threshold, cells enter senescence, characterized by dramatic alterations in morphology, gene expression, and metabolism. This phenomenon occurs across diverse cell types and has been documented in numerous species, with fascinating correlations to their respective lifespans. This discovery directly challenged Alexis Carrel's earlier theory, which incorrectly claimed that normal cells possess unlimited replicative potential. Hayflick interpreted his findings as evidence of aging at the cellular level. He suggested a direct correlation between cellular aging and the overall physical aging of organisms. According to this theory, the progressive accumulation of senescent cells throughout the body contributes substantially to the aging process. As these non-dividing cells increase in number, tissue regeneration capacity declines, leading to age-related deterioration and increased susceptibility to chronic diseases. Research suggests an ultimate Hayflick limit of approximately 125 years for humans. This represents a biological ceiling beyond which current interventions cannot extend human lifespan. For the physician, understanding this cellular phenomenon is crucial. It provides a molecular framework for age-related pathologies, from cardiovascular disease to neurodegeneration. While genetics accounts for merely 20 to 30% of an individual's lifespan, environmental factors and personal behaviors play a substantially larger role in determining how quickly one approaches this biological boundary. This highlights the profound impact of modifiable lifestyle factors on patient health span. At the molecular level, the Hayflick limit stems from a phenomenon known as the end replication problem. During DNA replication, DNA polymerases cannot completely copy the telomere regions at chromosome ends. This occurs because RNA primers, which initiate DNA synthesis during lagging strand replication, must be removed. Upon removal of the final primer at the 3' end, the newly synthesized strand inevitably loses nucleotides. Consequently, with each cell division, telomeres progressively shorten by 50 to 100 base pairs. Once telomeres reach a critically short length, the protective cap structure at chromosome ends becomes disrupted. The cell recognizes these uncapped telomeres as DNA double-strand breaks, triggering a persistent DNA damage response that halts further division. This protective mechanism prevents cells with potentially dangerous genetic abnormalities from continuing to replicate. Interestingly, cancer cells evade the Hayflick limit through the expression of telomerase. This enzyme extends telomeres and prevents their shortening. This enzymatic activity essentially grants cancer cells unlimited replicative potential, highlighting why Hayflick was the first to observe that predominantly cancer cells demonstrate immortality. Telomeres serve as protective caps at chromosome ends, functioning as a biological clock that determines cellular lifespan within the framework of the Hayflick limit. These specialized nucleoprotein structures prevent chromosomes from nucleolytic degradation, unnecessary recombination, and interchromosomal fusion. As cells divide, telomeres progressively shorten by 50 to 200 base pairs per division due to the inability of DNA polymerase to completely replicate the lagging strand, a phenomenon known as the end replication problem. When telomeres reach a critical length, the cell activates a DNA damage response, DDR, triggering cellular senescence or apoptosis. This biological mechanism helps explain why cells hit the Hayflick limit after a predetermined number of divisions. Critically short telomeres lose their protective function, becoming largely devoid of telomeric DNA and telomere-binding proteins. Telomeric regions are particularly susceptible to oxidative damage. The G-rich telomere repeat sequence is highly vulnerable to oxidative stress, which can directly damage telomeres and accelerate shortening beyond what occurs through normal replication. Unlike damage elsewhere in the genome, DNA damage at telomeres is resistant to repair. It therefore accumulates in cells, creating telomere-associated foci that persist even in non-dividing cells. Hence, both replicative senescence stimulated by critically short telomeres and senescence-like conditions induced by broken telomeres in non-replicating cells share continuous telomere DDR activation as their common underlying cause. Telomerase, a ribonucleoprotein complex consisting of telomerase reverse transcriptase, TERT, and telomerase RNA component, TERC, counteracts telomere shortening by adding telomeric repeats to chromosome ends. This enzyme's activity is strictly regulated in a cell-type dependent manner. In humans, telomerase is active during early in utero development, becomes inactivated in most adult somatic cells, yet remains active in germline, embryonic stem cells, and certain immune cells. Adult stem cells maintain lower levels of telomerase activity than embryonic stem cells. This is sufficient to slow telomere shortening and expand replicative lifespan, yet inadequate to prevent eventual replicative senescence. Telomere function depends not only on length, but importantly on structural integrity. Persistent DNA damage foci can form at telomeres, regardless of their length. The shelterin complex, consisting of 6 proteins including TRF1, TRF2, and POT1, plays a critical role in telomere protection. Dietary patterns exert profound effects on cellular aging through multiple pathways that directly impact telomere maintenance and the Hayflick limit. Recent research demonstrates that nutritional choices can accelerate or delay the pace at which cells approach their finite replicative capacity. This offers a powerful, non-pharmacological intervention for physicians to consider. The G-rich sequence of telomeres, TTAGGG, makes them particularly vulnerable targets for reactive oxygen species, ROS, damage. Oxidative stress creates 8-oxo-2-deoxyguanosine, 8-oxo-DG, within cells, causing DNA double-strand breaks, specifically at the GGG sequences of telomeres. Diets high in processed meats, refined carbohydrates, and high fat content generate excessive ROS that attack these susceptible regions. Excessive sugar consumption initiates a particularly damaging cycle. High intake of sugar and sweetened beverages elevates blood glucose levels, triggering oxidative stress in endothelial cells. This metabolic disruption has been observed to accelerate telomere shortening. Meanwhile, reactive oxygen species produced by dysfunctional mitochondria can directly damage telomeric DNA, triggering DNA damage response pathways and ultimately causing cellular senescence. A bidirectional relationship exists between inflammation and telomere dysfunction. Pro-inflammatory cytokines like TNF-alpha and IL-6 are elevated in chronic inflammatory conditions and have been found to inhibit telomerase activity, particularly the catalytic subunit TERT. Simultaneously, cells with critically short telomeres often enter senescence and develop a senescence-associated secretory phenotype, SASP, releasing inflammatory molecules that perpetuate a damaging feedback loop. This creates what researchers describe as a circulus vitiosus, where telomere dysfunction increases inflammatory signaling, which further accelerates telomere attrition. Chronic inflammation aggravates telomere dysfunction and cell senescence, while decreasing regenerative potential in multiple tissues. Certain dietary patterns show promising effects on extending cellular lifespan before reaching the Hayflick limit. The Mediterranean diet has been associated with longer telomeres and higher telomerase activity in peripheral blood mononuclear cells. In contrast, Western diets accelerate telomere shortening, pushing cells toward their Hayflick limit prematurely. Foods that positively influence telomere dynamics include fish, nuts, seeds, and olive oil rich in omega-3 fatty acids, colored fruits, and cruciferous vegetables high in antioxidants, green tea, red grapes, and tomatoes containing polyphenols. Conversely, dairy products, simple sugars, and foods with high glycemic load are associated with telomere shortening. Through these mechanisms, dietary choices directly influence how quickly cells approach the Hayflick limit, the theoretical ceiling of cellular divisions before senescence occurs. Specific micronutrients play crucial roles in maintaining telomere integrity and potentially delaying the Hayflick limit through distinct biochemical mechanisms. Their strategic consumption may influence how rapidly cells approach their finite division capacity. Physicians can advise patients on these targeted nutritional strategies. Folate and vitamin B12 function as essential cofactors in DNA methylation and nucleotide synthesis pathways. These B vitamins are required for converting homocysteine to methionine, which subsequently forms S-adenosylmethionine, SAM, the primary methyl donor for DNA methylation. When deficient, homocysteine levels rise, correlating with shorter telomeres in cross-sectional human studies. This association exists primarily because homocysteine accumulation increases oxidative stress and inflammation, accelerating telomere attrition. Vitamin C serves as a potent free radical scavenger that protects telomeres from oxidative damage. Studies demonstrate that vitamin C intake positively correlates with leukocyte telomere length and can reduce telomere shortening by up to 62% in cultured human blood vessel cells. Likewise, vitamin D supplementation, 2,000 IU daily, increases telomerase activity by more than 19%. A recent clinical trial showed that daily vitamin D3 supplementation significantly reduced telomere attrition over 4 years, preserving approximately 140 base pairs compared to placebo, effectively preventing nearly 3 years of biological aging. Vitamin E, available in 8 different forms, dramatically slows age-related telomere shortening, even when exposed to powerful oxidants like hydrogen peroxide. Polyphenols represent a vast group of plant compounds with over 8,000 identified molecules that enhance cellular antioxidant protection. Resveratrol exerts anti-senescent effects on endothelial progenitor cells by enhancing telomerase function and AKT phosphorylation. At merely 5 nanomoles per liter concentration, resveratrol significantly reverses senescence markers in later passage mesenchymal stem cells through upregulation of HTERT mRNA. Alternatively, curcumin inhibits telomerase action in tumor cells and induces telomere shortening, indicating its potential anti-cancer properties. Omega-3 fatty acids preserve telomere length through anti-inflammatory mechanisms. Research confirms that reducing plasma omega-6 levels while increasing omega-3s results in longer telomeres. A meta-analysis of 5 clinical trials involving 337 participants revealed a statistically significant positive effect of omega-3 supplementation on telomere length. Furthermore, omega-3 fatty acids reduce F2 isoprostanes and potentially increase clearance of neutrophils with shorter telomeres from circulation. Beyond individual nutrients, comprehensive dietary patterns exert distinct influences on cellular aging mechanisms and potentially alter how quickly cells approach the Hayflick limit, the theoretical ceiling for cellular divisions. Physicians can leverage this knowledge for patient counseling. The Mediterranean diet demonstrates remarkable effects on telomere dynamics. Individuals adhering to this dietary pattern exhibit longer leukocyte telomeres and greater telomerase activity in peripheral blood mononuclear cells. Even a short-term, approximately 1 month, Mediterranean diet intervention reduces the proportion of endothelial cells with shorter telomeres. Notably, higher adherence to the Mediterranean diet correlates with telomere length z-scores of 0.072 versus negative 0.038 for those with lower adherence. This difference corresponds roughly to 1.5 years of aging per 1-point change in Mediterranean diet score, with a 3-point change equivalent to approximately 4.5 years of biological aging. Primarily, the diet's protective mechanisms stem from its rich phytochemical content, which simultaneously reduces oxidative damage and inflammation. Alternatively, the Western diet, characterized by high intake of sugars, saturated fats, and processed foods, accelerates telomere attrition through multiple pathways. This dietary pattern promotes hypercaloric intake that generates reactive oxygen species and induces endoplasmic reticulum stress. Among mice-bearing tumors, Western diet consumption activated unfolded protein response pathways and enhanced tumor growth. Furthermore, this diet creates persistent low-grade inflammation, or inflammaging, which contributes to cancer induction and progression. Intermittent fasting presents another approach for managing cellular aging processes. This dietary strategy triggers a metabolic switch, where ketone bodies, particularly beta-hydroxybutyrate, serve as alternative fuel sources. Among its multiple benefits, fasting activates fatty acid oxidation, enhances intestinal stem cell function, reduces oxidative damage, and improves mitochondrial function. Clinical studies have documented improvements in cardiovascular markers, blood pressure, and enhanced cognitive functions. The molecular mechanisms involve regulation of AMP-activated protein kinase, sirtuins, and mTOR pathways, all linked to longevity genes. Both vegan and ketogenic diets offer distinct advantages regarding cellular aging. Vegan diets correlate with a 75% lower risk of developing hypertension and up to 78% reduced risk of type 2 diabetes. Throughout 18-week interventions, participants following vegan diets lost approximately 5.5 pounds more than non-vegetarians. Similarly, ketogenic diets show effectiveness for weight management, blood sugar control, and cardiovascular risk reduction. However, their combination presents challenges yet potential benefits. The vegan keto approach emphasizes high-fat plant foods like coconut oil, avocados, nuts, and seeds, while maintaining ketosis. Although no studies specifically examine telomere impacts of combined vegan keto approaches, each separately influences pathways affecting cellular senescence. The research presented throughout this video demonstrates the profound connection between dietary choices and cellular aging processes. Telomere maintenance, therefore, emerges as a critical factor in how rapidly cells approach the Hayflick limit. Evidence suggests that diet-induced oxidative stress, inflammation, and nutrient deficiencies directly accelerate telomere shortening, while making proper nutritional choices can substantially delay this process. Mediterranean dietary patterns are particularly beneficial for telomere preservation, extending cellular lifespan by reducing oxidation and inflammation. This contrasts sharply with Western diets that promote telomere attrition through multiple pathways. Certain micronutrients, namely folate, vitamin C, D, E, and omega-3 fatty acids, play essential roles in telomere maintenance through distinct biochemical mechanisms, including DNA methylation, antioxidant protection, and anti-inflammatory effects. Alternative eating patterns such as intermittent fasting trigger cellular repair mechanisms that potentially delay senescence. Both vegan and ketogenic approaches offer unique advantages regarding age-related cellular changes, though each comes with distinct considerations for long-term adherence. The ultimate implications extend beyond mere cellular longevity. Telomere preservation affects overall organismal aging, disease susceptibility, and quality of life. Although genetics contributes 20 to 30% to individual lifespan, dietary choices represent modifiable factors with substantial impact on biological aging trajectories. These findings underscore the remarkable plasticity of the aging process at the cellular level despite the theoretical ceiling imposed by the Hayflick limit. As healthcare professionals, we are uniquely positioned to translate this scientific understanding into actionable patient care. Integrate discussions about dietary patterns and specific micronutrient intake into your clinical practice. Counsel patients on the profound impact of nutrition on their biological clock and long-term health span. Consider telomere length as a potential future biomarker for personalized aging interventions. Stay abreast of emerging research in nutritional gerontology to optimize patient outcomes and promote healthy aging. If you enjoyed the video and found it helpful, don't forget to smash that like button and subscribe to our brand new YouTube channel. We're bringing you a dynamic mix of content across a variety of topics, so there's always something fresh, engaging, and insightful just for you. Your support means the world to us. It helps us grow and keep delivering exciting, high-value content. Stay tuned because the best is yet to come.